Okay, check, check. I think I have the right slides up. All right. Welcome in, everybody. How's everybody's February been staying warm? It's a little colder than usual. We actually have a winter this year. I feel like our spring will feel a lot more earned this year from what we've been through. Welcome to the Bird Study Group meeting of the Hamilton Naturalist Club. Here's just a little preview of the agenda tonight. We're going to talk a little bit about what this group is for those who are new. And who is new here tonight? Whose first meeting is this? Oh, very nice. Several new people. Welcome in. We're going to then talk about some recent sightings. Announce a little bit about Niagara Peninsula Hawk Watch. Not much to announce there, but just to tell people about it. We're going to have a very brief extra guest presentation on the agency survey that's being done, and talk a little bit about upcoming events, and then our guest speaker for the evening. And just for a reminder or notice for those who don't know, we do stream these meetings onto YouTube. We realized quite early after we started meeting in person again that it's much cheaper just to stream it straight to YouTube than to pay Zoom to do the same thing. And then we don't have to send out passwords, plus it saves everything right to YouTube because we were always uploading it there anyways. So we do that. And you can get this on youtube.com/@HamiltonNature. And you can follow in all of our past streams. And we did, before a pandemic, we actually recorded meetings often and uploaded them there. So there should be stuff back to 2018 or so, I believe. We're not the only naturalist club on YouTube. For example, I found out recently they own Sound Field Naturalists is there, not that far away from us. Just about 4 hours north of here. And their latest upload was 4 days ago, so they do have recent stuff too. So a little bit about us. We have been protecting nature since 1919, so we've been around quite a long time. We're dedicated to the study, appreciation, and conservation of plants and animals. And our meetings, like this one, and field events, are open to everyone. There's no requirement for a membership. But by becoming a member, you will get our monthly journal, The Wood Duck, and we'll be supporting our mission to protect nature for the Hamilton Study Area. And you might be wondering what the Hamilton Study Area is. Just a little map of it. It encompasses these lands here. This includes Hamilton, Burlington, and several other municipalities, and also the lands of Six Nations, the Grand River, and Mississauga's Credit First Nation. So we consider this to be kind of our focus area for study. These lands have been traditional territories of various Indigenous peoples, including the Erie peoples, Adirondack, Huron-Wendat, Haudenosaunee, and the Mississaugas. And there are many treaties that cover this area. For those who aren't aware, there's quite a few on there on the screen. And additionally, these lands are covered by the Dish with One Spoon Wampum Belt Covenant, which is an agreement between the Haudenosaunee Confederacy and the Anishinaabe to share and care for the resources around the Great Lakes. As the agency, we actually protect 11 sanctuaries, most of which are in the HSA, but a couple aren't. They are listed here, and the newest one was Pine Hill in 2025. This is something that donations help us do and help us maintain, which is the even more important part. Acquisition's one thing, but being able to do restoration and maintenance work is a whole other thing. For the bird study group specifically, though, we have these group goals to keep our minds on the birds beyond what we see and hear in the field, so things like our speaker tonight. Bring awareness and amplify efforts, initiatives, and projects that are having a positive impact. Allow broader conversation about bird ecology, threats, efforts, and trends and predictions. To bring the average person and the expert birder into the same space. Now, I usually try to make a little bird of the month slide. I do have one, but wow, there wasn't a lot to choose from this last month. There hasn't been a lot since the last presentation. But we did have a Ross's goose show up at the Wilkes Dam in Brantford, found by Elliot Holtveur. And these are the photographs by Joan and George Sims, lifted off to eBird, a few different photos of this bird. And just to show that there's not a lot of like notable HSA sightings, here's just, what I could find. And some of them I had to kind of hunt for a bit. There was, of course, this Ross's goose, the yellow-rumped warbler at Brant-CA, although I think there's probably a couple of others out there. The Eurasian teal that's at Windermere might still be there. I'm not sure if it's still there, but I know at the end of February or end of January it was still there. There was a harlequin duck at Tannery Park in Oakville, merlin that I spotted at Mount Hamilton Cemetery. It might have been some other merlin. Actually, we had a merlin on our walk at Gage Park too. I forgot about that, so that should be in the list. Northern Shrike at Paddy Green Road, and there was one in Saltfleet too. Gray catbird and fox sparrow were both on the Anishinaabe Wazidawin RBG Arboretum Trail. Pine siskins, they were all over the place. I haven't seen very many this year. I saw some right after New Year's and then not much else, but I've heard stories of people having huge flocks in their backyards and stuff like that, so that's kind of neat. Yellow-bellied sapsucker, this kept coming up on eBird Rarities like almost daily. I think they're kind of back now, it seems like. I don't know a lot about the history of their occurrence in the HSA very much. I just know they've always been kind of uncommon to rare often, and they seem to be a lot more common this winter. Red-shouldered hawk has also popped up in a lot of places. I know I heard one at McMaster Forest a few days ago that was doing its classic call that sounds like it's it sounds like it's a young hawk in distress, but it's actually that's what their normal call is. Also, just there's a couple requests for help. One is we'd love some help for the refreshments that you have out there that many of you have enjoyed. We'd like some more help on that refreshment committee. So I said I'd remember when I saw this slide because I didn't have a chance to put it in here, but get to your thing before mine. But yes, we could always use more help with that, and it's great food too. So greatly appreciated.
Ellen and I, we are doing it, but 2 meetings a month.
Yeah.
It makes it difficult. So
Yeah.
it would be nice to have more people so that we don't have to do it every.
Yeah, exactly. What you're saying is just because there's 2 meetings a month, that's a fair bit. But if we can get more help, then maybe it's only 1 meeting a month you need to do this, right? So, also, we're looking for help in coordinating a accessible birding locations list. We've done location lists in the past, but it would be with eBird and stuff like that these days, it's very easy to find where the popular places are. But when it comes to curating lists about how accessible something is, such as it's on the bus route, may not very many like uphill, downhill, incline, decline, that kind of stuff. Places that are good for long watches and stuff like that. What we're not looking for is for people to just send me a bunch of locations. What I'm actually looking for is for someone to send those locations to and them to make that list. So that's what we're seeking right now. And if somebody would like to come forward, that would be amazing. You can email me at the email address there, or just reach me afterwards as well. And it being soon to be March, the Niagara Peninsula Hawk Watch, said that too fast, is coming up. They always need volunteers. I just went and kind of lifted this off their website, assuming they're always going to need more volunteers because they need people to be counting every single day. And I'm assuming that the weekdays are going to be the tougher ones that for them to fully book people in. So, but also if you're just completely new, go ahead.
Sometimes Sundays too.
Sometimes Sundays too. Okay. All right. Good to know.
And we need more volunteers. Last year I did 5 times, and 3 days I was all by myself. It's nice to have a second person.
Yeah.
Too.
Yeah, and that's also something that's important as well. Like maybe a day is covered, but it's just one person there. And it's always good to have 2 or 3 because more eyes in the sky or on the ground, wherever, always handy. Plus mentorship opportunities. So even if you don't know what it is you're looking at, there might be a person next to you who does know, and then you can learn from them. So it's a great learning opportunity. I'd like to take a few minutes here to call up a special guest presenter to talk, or a couple presenters to talk about the Hamilton Naturalist Club's strategic planning survey, which is a really important thing we're working on, and these folks are going to explain it.
Can I switch off to?
Oh, right. Sorry. PowerPoint.
So while we're getting that up, I'm going to introduce myself. I guess
There you
I
go.
should come over here to the
There you
microphone.
go. You can use this if you want to. Forward, backward if you want.
Oh, okay. Thank you. My name is Sonia McDonald, and I am one of two principals with a group called Civicplan. We're based here in Hamilton, and we're helping the Naturalist Club with a strategic plan. My colleague, Paul Shaker, is with me, and we're just going to take a few minutes. I know some of you have already heard this spiel, but we're back again because we really, really, really need your help for this. So, as we heard, the Naturalist Club's been around since 1919, and in its 107-year history, it's never had a strategic plan. So, good time to come for the first time because you can help form the organization. So what we're here to do is to help do that from the membership up. And the idea really is you all have experiences with the club, you all have different activities groups and so much great information about how it works and how it works well and what you want to see more of. So we're here to help coalesce some of that and then organize that into a strategic plan for the next 5 years or so. So what we are going to start with, obviously, is what we're hoping to do with the plan is to see that everybody's really on the same, so either on the same page or not, but get a sense of where people are coming from. We want to confirm your existing priorities and set any new priorities if those come out in what we hear from you. And then we hope that the plan is going to inform all of your future actions and directions. So the process is, as I mentioned, we want to start from the ground up. We want to start with a survey of all the members and volunteers to gauge how they view the club and its future. That will then lead into a workshop with the board of directors, and we'll facilitate that to review and confirm any of the goals and priorities for the organization. And the workshop will be informed by the results of the survey. So that's why we need some really great input from the survey to then inform that workshop. And all of that will be coalesced into the strategic plan, which will really feed from those outcomes. Now, the survey is available. I'm going to turn it to Paul, who will explain the survey. And I am going to say this, and he'll say the same thing. It is long, and I know it's long, but it's because you do so much. And we really, really would love for you to take the time, go through it, give us your real thoughts, and that would be really, really helpful. And a lot of you have already done it, but we're going to keep asking for folks to continue.
So just in terms of survey structure, to see what we're looking for here. And I should say, first of all, the survey was developed with, there's 2 phases. We worked with a sort of a subcommittee of the board to do a sort of preliminary run through, and then it was run past the whole board for their feedback on structure and if it was going to be capturing all they wanted to see and solicit feedback from membership and volunteers. But the survey structure, and there's a link to it, well, the next slide I'll show you in a second, but basically what we're going through here is, first of all, just introduction and background to see, there's varying people of different backgrounds and levels of involvement, so they may not know some of the history of the club. So just putting a bit of background on the table. And then really looking at mission and goals of the club, because some people don't even know what they are, and so we want to put them forward to explicitly ask membership what they think in terms of whether that aligns with what they feel the club should be doing, what it does, or maybe does need to be altered. So there's a section on the mission and the goals. And then we dive into the projects, activities and events, and the sanctuaries, really all this sort of the activities of the club. So a number of projects are put there, and then for each of those sections, actually, we look into whether you participate or not. The survey is structured so if you are a participant of an activity on the survey, you can select yes or no, and then if you aren't, it will simply ask, why don't you participate? And that's the end of that sort of section for you for that project. But if you do participate, it opens up more questions about everything from how you participate, what you enjoy about it, what could be improved, all sorts of elements, and how well you think those activities align with the mission and goals of the club. So we go through a whole series of the projects, the activities and events, and the sanctuaries. And then we go to another section, and what came up with the board here, this is, since we're going to the membership, this is a great opportunity to get all sorts of information to really help the board decide and plan all sorts of things, not just into strategic plan, but other things. So other topics, communications and outreach. You know, how's the club communicate right now? Is it effective? What could be improved? Methods of outreach, how that is working. How do you folks solicit information about the club, and could you be communicated with in a better way? So all sorts of questions like that we've included. And then even for the Wood Duck, the magazine, both online and the physical copy, the folks there wanted to dive in and ask questions for their purposes about the use of that in publication. You know, everything from frequency of readership, satisfaction, all sorts of things of that nature. So these are wrapped into this survey as well. And finally, we have a section on demographics, because one direction from the board was they wanted to see what is the makeup of the membership? How diverse is it? You know, everything from ethnicity, age, terms of background, all that sort of thing. They want to get a sense. Is it representative of the population? They want to get a good picture, but what is the membership and where the strengths and what could be improved upon? And even we get into things like, how do you come to attend meetings such as this one? How do you get here? Physically, what mode of transport do you take? Is it convenient? You have the online option, but wanting to get a real sense of how people interact with the club. So that's the different sections of the survey, and as Sonia mentioned, is lengthy. There is a physical version as well as an online version, and I mean, folks here, that's the web address you can use to get there, or you can even click on the QR code. But we do have physical copies if folks want them. And I'll just a plug for why the online version is better for a few reasons. One thing, you're saving paper. This is a long survey. We try to limit how much we're using paper. But as well, what I mentioned about that idea of activities and events, the online survey really allows you to dive deep in just the things you really participate in. And it, in fact, shortens it for you taking that version. The paper version, we print out all the questions for all the activities, so it's just that much bulkier. But that's the survey outline. It's been open for a couple of weeks now. We've had some good feedback, but we hope to have it for at least another 2 weeks. But I've said to the board too, this is such an important thing that if you need a few more weeks into sort of getting to March break, by all means, we want to keep it open for that. So for all those who've taken the survey, thank you. There is a prize draw actually of up to $200, I think, dollars, which after you finish the survey, you can then put your name anonymously from your input into a draw for $200. But for all those who have taken it, we thank you. For all that haven't, please take note of the web address and take the survey. And we look forward to the feedback. Thank you. Oh, any questions? Yes.
Can I just ask a question?
Sure.
You mentioned communication. I'm a long-time agency member, but I can't recall seeing any email or notification that this survey existed. Okay, now I'm guilty of skimming sometimes, and I might have missed it, either here or in the Wood Duck, but I haven't seen anything on Hamilton Birds or Google Groups. Rob might be able to inform better on that one, but
The Google group is not run by the club.
I would gladly do the survey because I think it's important.
Yeah.
This is the first I've heard of it.
Oh, okay.
Yeah.
All right, so you might need to be on the newsletter for the Hamilton Naturalist Club, because the Google groups are not run by the club, they're run by other people. So I could see about getting out there, but then we have the problem of, I'll talk to people and see if that would make sense or not. But yeah, you should be getting monthly emails from, I think it's the president's message or something like that, and all that. So it should have been in that, is where you should be
And
seeing.
it's coming in the Wood Duck.
And it's coming in the next Wood Duck as well.
It'll be out next week.
Next week, okay.
Okay.
Yeah, that's part of why we have it here in the meeting too. We want to make sure we hit it every, because I know it's easy to miss an email, very easy.
Find out now than be in the
Yeah.
dark forever.
Yeah, no, we want to be very thorough. Yeah,
Excellent.
yeah, like you said, we want to be very thorough in this. This is why we haven't necessarily put a deadline yet. It's more of a goal-oriented thing where if we are not seeing enough results, we're just going to push even harder to get even more, like ideally every single member will fill it out, is actually really the goal. But,
Exactly. Yeah.
yep,
Hey Rob, you can do the Google group on the first of the month.
yep. We'll talk about that and see what, because I could probably get a special exception too for that. So,
The open forum was specifically what I was thinking about.
yeah,
With respect to Hamilton, there's a Google group.
yeah. So that's something we can chat about. Yeah. Do you have any other questions here?
Anyone else?
Thank you for that.
Great,
Okay,
thanks.
thank you.
It's always good to have more feedback on that. Let's see. And play. Backup. All right. So before we get to our speaker, just a little bit about upcoming events as well. But if you end up with any more questions, these folks are here for a little. Are you guys here till after the presentation?
Contact me too.
You can contact Jen as well, or reach out to me and I'll find the right person. So, the West End of Lake Ontario KBA survey, as always, taking place second Saturday each month, up through April. 17 areas to cover. You can email Chris CMTrain at cmotherwell.com for that. So we have some events coming up, such as this one on Saturday, March 7th. Stories of the Iroquois Sandbar with John Terpstra. So I think I had this on in the slide earlier before you all came, like everyone came in before we started, but you can find this on the HNC website as well. It's a limited event, so you'll need to email fieldevents@hamiltonnature.org if you're interested in this one. But it's also listed on the agency website events calendar. Trivia for Nature. This one's a little farther off, but we want to get it out there beforehand just to, have you save the date. April 18th is an annual fundraiser for the club. This one is at 29 Park Street West, Dundas. Is that a particular venue?
St. Paul's United.
St. Paul's United? Okay. And, $15 per person. Cash bar, bring your own snacks, 50-50 raffle, 7 to 10. In general, it's general trivia questions too. I always have this impression that it's all going to be nature questions, but it's general trivia. Yeah, so it's for everyone. Another field event, March 15th. Paul O'Hara and Julia Hitchcock are leading a relaxed medium-pace walk along the Escarpment Rail Trail near Wentworth Street South. So we'll be enjoying the trees, chat about the winter tree and plant identification, traditional uses, and disturbance vectors for urban woodlands. The trail's flat, gravel paved, a few muddy spots. You can RSVP with field events at hamiltonnature.org. And a couple of the next meetings. So the March meeting isn't that far away because of how calendars work. It's actually only 2 weeks away. So general meeting will be intersecting applied science with community climate action. So this has to do with trees in the St. Catharines downtown business improvement area, I believe. And these people, Lindsay and Sean, came with the question, how do we bring more trees downtown? And through their research, they developed a method to accurately and responsibly measure and monitor the trees as green infrastructure and natural assets. So I'm definitely looking forward to this one because it's always been something I'm curious about. So that's Monday, March 9th here, 7 p.m. Doors open as usual, 7:30 for the meeting start, and online if you can't happen to be here. And the next month's bird study group, we have Ben Walters from Fanshawe College, I believe, or no, Fleming College, actually, from Ontario's Kirtland's Warbler Project. This is one I've been wanting to get in here for a while, is to talk about Kirtland's warblers, especially right before the season starts. This is about stewardship and recovery, and it's an emblematic bird of Michigan, but there's a lot of birds that come into Ontario, and there's some new populations forming, I believe. But this is something that Ben will be speaking to. It's been a rare bird, of course, in the long history in Ontario, and various conservationists from academic institutions, nonprofits, and industry agencies have collaborated to establish Ontario's Kirtland Warblers Working Group. So it'll be interesting to hear about all that. But I would like to invite tonight's speaker up. Dr. Jolene Giacinti is going to be talking about avian influenza, understanding the virus behind the headlines. And come on up, and we'll just take a second here to set up the slides. Let's see. All right, and this should work for you.
Perfect.
Good evening, folks. It's great to be here and to hear about all of these awesome events that are happening in and around our city. So I'm just going to start by introducing myself. So I'm Jolene. I'm a veterinarian and I'm a scientist, and I work for Environment and Climate Change Canada. We'll talk a little bit about what Environment and Climate Change Canada is in a couple of slides if you're not familiar. But I study wildlife health and wildlife disease, and the last few years of my life have been really dominated by avian influenza. For those of us who are out and about and seeing the impacts that avian influenza has had on our wildlife species, it's been a little bit hard to ignore. So what I'm going to be talking to you about today is a little bit about what the virus is. There's a lot out there and the language can get complicated, and so I'm just going to break it down a little bit. Talk about what we're doing in Canada to understand the impacts of the virus and how it's changing over time, some of the questions we still have and what we're doing to answer them, and then hopefully along the way I'll answer some of your questions as well, but there should be time at the end to ask questions that I don't cover. And please feel free to interrupt me along the way if I've said something that doesn't make sense. Yell it out. I'm happy to clarify. I'd rather stop and explain in the moment. So I'm going to start by playing this video. Hopefully it's going to play. Let's see. Oh, I pressed the wrong button. Okay, there we go. So this is just an example to get us started. So this is a banding station at Hawk Cliff. You may or may not be familiar, but this is just an example of one of the really amazing places and some of the amazing people that I get to work with as part of my job in understanding the virus. And we'll talk a little bit more about some of the work we're doing at Hawk Cliff related to avian influenza throughout the talk. So let's get started. So I said I was going to talk briefly about Environment and Climate Change Canada, and so this is where I'm going to do that. So it's a branch of the federal government for those that aren't familiar, and it actually has a pretty broad mandate to preserve and enhance the quality of our natural environment. So there's a lot of different pillars of work that happen at the agency. The work that I do falls into the pillar related to conservation, protection, and management of wildlife and habitat. So that area highlighted in blue there on the slide. And specifically, we have a mandate to conserve and protect migratory birds and species at risk. And so that's where my work comes in, because migratory birds and species at risk are both impacted in association with avian influenza virus. And this is a reminder for me to just let you know that I've put QR codes throughout the talk where I think there's additional resources or guidelines that you might find interesting. So please feel free to make note of these along the way, but I'll bring them back at the end of the deck as well if you miss them. Okay, so what is avian influenza virus? I'm going to back up just one step from that because it's actually part of a larger family of viruses that we know as influenza viruses, and that'll be familiar to you because humans get influenza viruses as well. And so avian influenza viruses are part of what we call influenza A viruses, but there are other types of influenza viruses, and they each infect a different range of species, which is what's indicated by the circles on the slide there. But influenza A viruses can infect the largest number of different species, so that's why this circle is the largest, the one in yellow here. Avian influenza viruses are a subset of influenza A viruses that are most adapted to birds, and that's why they're called avian influenza viruses, but they can actually infect other species as well. So don't worry, we're not going to have a virology lesson here, but this is just, we're going to talk about a couple of the ways that we refer to avian influenza viruses because there are a lot of different ways and it can be complicated. And so this is one of the most common ways, and it's what we call subtype. So you may have heard H5N1, and so we're just going to talk briefly about what that means. So this is a picture of what a virus particle might look like. Of course, it's sort of a cartoon depiction of it, but there's two surface proteins that I've circled on the slide there that are really important for the virus to be able to infect a cell and a host. And those are the HA and NA proteins. And we refer to these by number based on the specific subtype of the virus. So when we say H5N1, we mean type 5 of the H protein and type 1 of the N protein. And there's a whole bunch of different combinations of subtypes that are out there, like H12N8. But for the purposes of what we're talking about today, it's H5N1. So if you've heard that term and didn't know what it referred to, hopefully that helps a little bit. Another way we refer to these viruses is by how much disease they cause in domestic poultry. And so we refer to this as pathogenicity. And so we refer to low pathogenicity and highly pathogenic viruses. So what does that mean? So what we're seeing in the picture here is what low pathogenic avian influenza viruses look like in a domestic bird. So they tend to be sort of localized infections in the upper respiratory tract, in the intestinal tract. So you can see the tissues that are sort of in the purple color there. We tend to see mild clinical signs in these birds associated with these viruses. When we're talking about highly pathogenic avian influenza viruses, it's more of a systemic infection. So it can infect all of the tissues in the body. See all of the purple tissues there. It tends to cause really significant illness and often death in these birds. But what's really important is that we refer to this based on the disease in domestic birds, not in wild birds. So we're going to be talking about the impacts in wild birds throughout the talk and what it means for wild birds. So I'm going to start talking about what low pathogenic avian influenza looks like in wild birds. So it's actually really common in some species of wild bird, and particularly waterfowl and specifically dabbling ducks. And so infection is common. So we commonly see low pathogenic viruses in these birds, but it doesn't typically cause any obvious clinical signs in these birds. So they're what's considered a natural host for these viruses. And there's a long history of them circulating in North America. So we've had people studying these viruses for decades now. So we've actually learned quite a bit about them. And what we know is that the seasonal movements of migratory birds from their northern breeding grounds to their southern wintering grounds is really important for understanding how these viruses move over long distances. So we can follow the viruses along these continental migratory flyways, which in North America we refer to 4 main ones, which are pictured in that diagram there. We also know that there's more virus on the landscape. We detect it more, typically in late summer leading into fall, and that's because this is when migratory birds gather in large numbers preparing for migration, and there's also more juveniles in the population that are immune to the virus. So they've never been exposed to it before. They don't have an antibody response generated that can help protect them from the virus, and so we see peaks in virus activity at this time of year. But what's important here is that when we say wild birds are natural reservoirs for avian influenza, that's only low pathogenic. Highly pathogenic avian influenza is not, we don't have a history of it in North America. Our birds are not natural reservoirs of these viruses. It actually originated in poultry. And we'll talk more about that on this slide. So this is just a brief history. So the viruses that we're talking about today, the H5 highly pathogenic viruses originated actually in the 90s in China. They were largely a poultry virus at the time, but over time, with repeated spillover events from poultry into wild birds, the virus was able to adapt to wild birds, and we saw it increasingly move to new locations, Europe, Africa, Russia, but not North America until 2014. So that was our first detection of a highly pathogenic avian influenza in North America. It remained a pretty localized outbreak at the time. There were poultry detections in Canada and more in the US, and a handful of wild bird detections. We didn't detect it after 2016, to the best of our knowledge. It was eliminated from our populations. However, in the background and in the interim, it was still circulating in other parts of the world, and the virus was changing throughout this time. leading us up to 2021, when we had another introduction of a highly pathogenic avian influenza into North America. And this time things were quite a bit different than that time in 2014, and that's what we're going to talk more about today. So the first question is, well, how do these viruses get here? If they're in Europe and they're in Asia, how are they getting into North America? And you folks probably know this, maybe better than I do even, but in addition to those continental flyways, there's also transoceanic flyways. So there are some species that travel quite long distances, or they have stopover sites that interface with birds that are then going to come into North America. And so this is thought to be the primary way that these viruses are introduced into North America. And we have flyways and potential for these introductions on both coasts, so the east and the west coast. So if we look at what introductions we actually had, we had our first introduction in November 2021 in a great blackback gull in Newfoundland. And just to clarify, that's not to say it's this bird or even this species that is the one that brought the virus over. This is just the first bird that we detected the virus in. But it wasn't just this one introduction. So we actually had multiple other introductions of what we call Eurasian highly pathogenic viruses. So it's not just one virus on the landscape. There's actually a number of different viruses that were introduced. And so if we look at the detections in the first year following these introductions, that's what this video is going to show. We can see the detections starting on the East Coast. We start to see some detections in Ontario, and then you can start to see the detections on the West Coast come in. It moves into the prairies, and then we start to see detections into the north. And the different colors of these dots on the slide here represent different taxonomic groups of birds. And so this really very quickly was an indication that this was going to be a different context than what happened in 2014. Many, many more detections across multiple different species groups, and in some cases we were seeing large-scale mortality events in our wild bird species, and we'll talk more about that. So pretty quickly, we needed to better understand what was going on, track it, how it was changing over time, and how are we going to respond to it? What are we going to do? And so Canada actually has a number of different activities happening across the country to understand and respond to these viruses. I'm going to have a couple dabbles of some of these other activities throughout, but primarily I'm going to be talking about our surveillance activities in the country. And what is surveillance? So surveillance is really our way of building a picture of what's going on and how the virus is behaving in wildlife. So we're trying to understand where it is on the landscape, where it's moving, what species it's infecting, and what it actually means for our population. So what are the impacts on our populations? This information is not just important for wildlife, though. It's also important for our colleagues that work in the agriculture sector. So domestic poultry has been heavily impacted by these viruses as well. And so the data that we generate through our surveillance activities in wildlife are very relevant to our colleagues that work with poultry as well. But then also for human health, because there are and there is the potential for these viruses to infect humans. Thankfully in North America, those cases have been very rare so far, but it is something that we always need to keep in mind. And so our colleagues in public health use our data to help understand what the risk is for people and Canadians. So before I start talking about some of our data, I just want to acknowledge that our surveillance activities in Canada are only possible because of a large collaborative effort across the country. So we have very, very many partners across the country that help us do these surveillance activities. Federal government agencies, provincial, territorial government agencies. We have partners in academia. We have partners, we have NGO partners. We also have, of course, the Canadian Wildlife Health Cooperative, who we work very closely with on this pathogen. So I just want to acknowledge all of the efforts from all of these very many partners. So how do we do surveillance? Well, there's two primary complementary sampling approaches that give us different kinds of information. So if you remember when we talked about what highly pathogenic avian influenza looks like in poultry, now we're talking about what it looks like in wild birds. So we actually have two different things we can see. We can have wild birds that get sick and die. We can also have wild birds that get infected and look apparently healthy and recover from infection. And so we need to target those two different groups using different strategies because they give us complementary information about what's going on. So if we start with sampling of sick and dead wild birds, this is largely led by our colleagues at the Canadian Wildlife Health Cooperative and also the provinces and territories. And it relies very heavily on folks like you in the room who are out on the landscape, you see a sick or dead wild bird or wild mammal, and you report that. And that gives us the ability to go and investigate the cause of death for that individual, and that's really what we rely on to study the impacts of these viruses in wild birds. So you guys play a really important role in that surveillance. The other important piece comes from the sampling of apparently healthy birds, and the way that we do this is through bird banding activities that are ongoing, and also through hunter-harvested the sampling of hunter-harvested animals as well. And this is really important because these are infections that would otherwise go undetected. These birds look apparently healthy. So if we're not actively looking for it, we're gonna miss those detections. And the reason this is important is because the birds that are gonna get infected and live are likely the ones that are gonna be moving these viruses over long distances. And so it's important that we understand what's happening in those species. So these approaches really work together. And remember, if you're going to get infected and recover from the virus, typically it stays localized in just the upper respiratory tract and the intestines. And so when we're sampling for this virus, we actually take a swab of the mouth and we take a swab of the cloaca. And so these photos are just giving you sort of examples of what that looks like. We pool those swabs together and we send it to the lab for testing. And I do just want to note that when we do sample these birds, we do it in such a way that they're able to be released after and go about their normal activities. So we just sort of add it on to regular banding activities, and it's in no way harmful for the birds for us to do this sampling. But looking for the virus in this way is only really part of the story. So if we look at this graph, if a bird is going to get infected with the virus, it's actually going to only have virus present for a short period of time. So if the bird's going to get infected and it's going to die somewhere in here, right, where the virus is at the highest levels they're going to be. And so it's going to be pretty easy for us to find that virus. But if the bird's going to go on and live, it's going to mount an immune response, which is going to quickly decrease the amount of virus that's present. And so we only have a short window of time, days usually, to detect the virus in that live bird. So what we can also do is look for antibodies, and that gives us a longer window of time through which to gain information from these populations. And sampling for antibodies requires taking a blood sample. And so these photos show some examples of that. So in larger bodied species like dabbling ducks and geese, we collect the blood into these vials. But then of course for smaller bodied species like this jay photoed, we collect smaller amounts on filter paper. And again, this sampling happens in such a way that the birds get released after and are able to go about their normal activities. But this data is really, really important because what it does is it helps us understand how many birds were exposed and survived from the virus, which helps us understand what the immunity is in that population and what that population's risk is if the virus were to be reintroduced to that population. So this is really important information for us as well. So if we look at what these activities look like across the country since fall 2021, our first detection, we can see the scale of the surveillance activities that are happening. So we've swabbed more than 68,000 wildlife across the country. and sampled more than 14,000 individuals for antibodies. And when we combine the virus and antibody data, we can start to build a much broader picture of what's going on and how it's moving through wild bird populations. So if we look at that data over time, so what we're looking at here is a time scale. So you can see the years across the bottom, and the number of detections are shown by the bar graph. So both the red and orange are showing likely detections of highly pathogenic avian influenza virus. And we can see that in this temporal pattern, how the detections rise and fall over time. So we can see that the size and timing of peaks does vary over time, but the virus has persisted across these years, which means that it's sort of here to stay, right? It's not like in 2014 where we weren't able to detect it after a short period of time. It really has persisted for a number of years. What we also see is similar to low pathogenic avian influenza viruses, we do start to see increasing activity in the fall prior to that migration of migratory waterfowl. But interestingly, for highly pathogenic avian influenza viruses, we see activity even into the winter, which is pretty atypical for what we would expect. And so you can actually see increasing activity through November, December, and even into January. So if we look at the detections in southern Ontario, just since January 1st of this year, we can see that the virus is still very much present on the landscape. So these are our detections in wildlife in the province just over the last couple of months. And so it is really important that we don't just think about this virus as being present in the fall when birds are migrating. It can still be present at other times of the year as well. So another important question is what species are impacted by the virus? So this is just a list of the species that we've detected with highly pathogenic avian influenza in Ontario. I'm not going to go through them one by one, but you can see that there's some patterns. Oh my goodness, I really jumped ahead there. There's some patterns in the data. So a lot of different raptor species, a lot of waterfowl species, not so many passerines, largely corvids, and then sort of a handful of other that I've just grouped as other. But if we look at this in terms of what are these patterns showing us, it's actually not evenly dispersed across which birds are apparently healthy after infection and which birds actually get sick and die following infection. So this figure demonstrates sort of some of the patterns that we're seeing. So if we look at live and harvested wild bird detections, we see that it's largely waterfowl, but in particular those dabbling ducks. So similar to what we would see for low pathogenic viruses, right? These birds seem to be particularly resilient against these infections, and these are likely the ones that are able to move at larger distances. When we're looking at what species are getting sick and dying associated with the infection, it's quite a bit different. So we do still see waterfowl, but largely geese, not dabbling ducks, and then we have diving and sea ducks as well. So it's not that we never see dabbling ducks die from this virus, it's just much less common than other species. We also see a number of raptor species. Again, corvids seem to be particularly susceptible, and then a whole host of seabirds, and in particular colonial breeding seabirds.
I have a question.
Yes.
So in the winter now, we have a lot of sea ducks coming in.
Mm-hmm.
So what is happening with those?
Yeah, so that's a great question. So the detections we're largely seeing right now in Ontario are geese and corvids. So those, and raptors actually, those tend to be the most dominant species that we're detecting. That's not to say other species aren't dying, we may just not be detecting them, right? So with sea ducks, sometimes we can have a harder time if they're dying out on the water and nobody's able to detect them, right? So I'm really glad you brought that up though, because this is a great time to say that all of our data is based on, there are some biases associated with how we collect the data, right? So we only know about what gets reported and what we're able to detect. So that's a great point, and thanks for the question. The other thing I want to note is that when we're doing surveillance of sick and dead wildlife, if there is a larger scale die-off, we're not testing every single one of those birds. We're typically collecting the bird carcasses and mammal carcasses that are in the best condition so we can get the best diagnostic result. But we're not testing all of them. So these numbers don't reflect the scale of some of the mortality events that we've been seeing, but I'm going to show some of that data in a couple of slides, just so you have some examples. Yes?
I've actually got two questions.
Okay.
If this is the appropriate time.
Yeah.
I noticed in your introduction, you talked about partners in respect to this
Yes.
study. And I noticed you talked about Hawk Cliff, but I also noticed Bird Studies Canada in there.
Mm-hmm.
You showed examples like, for instance, live ducks harvested or from hunters. Then you showed a picture of a blue jay.
Mm-hmm.
Is that where like organizations like Bird Studies Canada and Hawk Cliff come in? These samples are taken during banding?
Yeah, so that's a great question. So the Jay photo was actually not in association with those organizations. So that was a specific project where we actually went out and sampled at people's bird feeders. But yeah, in order to target and get samples from the diversity of wild bird species, we do have to work with these different partner organizations, because Environment and Climate Change Canada, for example, doesn't have programs on the ground for all of these different species, right? Largely, our banding programs are in waterfowl. So yes.
And the second part,
Mm-hmm.
the second question was, I noticed in the slide you showed previously about the species that were affected.
Yes.
You did mention corvids.
Yep.
But I noticed that gulls weren't in that category. And yet, in my mind, gulls and corvids eat carrion.
Mm-hmm.
Is that the connection? And what's different about gulls that you know of that makes them less susceptible?
Yeah. So that's a great question. There's several parts to that question. And so what I will say is that what we're learning is that species like raptors and other species that are predators or scavengers are particularly susceptible, because if a bird has died of avian influenza, if you remember the image that I showed, the virus is all throughout that carcass, right? So when it's scavenging or predating, let's say it's predating on a sick animal, it's getting a very large dose of that virus, and we think that's why raptors and other scavengers and predators are particularly susceptible. For species like seabirds, they're particularly susceptible at certain periods of the year when they're in high density. And so colonial breeding species, that breed really, really close together, they're pooping in close proximity, or they're grooming each other, the virus can transmit back and forth more easily. So it really depends on the behavior of that species and the ecology of their environment and where the virus persists in the environment that makes a species either higher or lower risk. So what you would note actually in this slide and the previous looking at species is that songbirds don't feature heavily in either of those slides. And it's not that there's no risk for songbirds, it's just that they really aren't commonly detected with the virus. So there has been some detections in the US in some songbirds like starlings that are present on a poultry farm that has the virus, and there's lots of virus in the environment and they get exposed. But largely songbirds, the ecology and their behavior just puts them at lower risk. They're not likely to interact with this virus as much. So there's a lot of different factors at play there. Have I answered your question? Because there were multiple parts to that.
I mean, to me, there's a commonality, like when you talk about colonial nesting birds,
Mm-hmm.
gulls are certainly, all you have to do is go down to the harbor. and
The
see
gulls,
them.
yeah.
And yet they seem to be relatively immune. I mean, songbirds to me would be more a matter of exposure.
Mm-hmm.
Is
That's
that
right.
fair to say?
They're likely just exposed to the virus less based on, where they nest and what their activities throughout the day.
Gulls are even tougher than we think they are.
So gulls, we do see gulls. Gulls are interesting. We do see gulls dying from the virus, but not as many as some other species. And gulls actually have a history of exposure to low pathogenic avian influenza viruses as well. And we think there's an element of immunity that they have when they're exposed to these related low pathogenic viruses. So dabbling ducks is another good example. Gulls is another good example. So if they have these previous exposures, it makes them more resilient to new exposures to new viruses. So we think that gulls actually can play an important role as well. But they're kind of, we do see some die, but some also do definitely survive infection, for sure.
Thanks for your answer, and I know we have a curfew here, so I
We'll keep going. But it raises a good point that these individuals that are sick and dying, this is where it's really, really important for us to collect those blood samples to test for antibodies. Because if all we're detecting is birds that are sick and dying, we don't know how many of those species are getting exposed and surviving. And that's really important for us to understand, to know what the risk is for these populations of species that are getting impacted, right? So some of the work we've been doing at Hawkcliffe is exactly that. So in association with their banding activities, we've been sampling any raptors caught using swabs and blood samples to test for antibodies. Okay, so I'm going to give a couple examples where we've seen some larger scale mortality events associated with the introduction of the viruses. So we're going to go to the East Coast first. In the northern gannet, and these are probably one of the species that were most heavily impacted. And if you're not familiar, northern gannets are a long-lived colonial breeding seabird. They breed on these cliffs, and so this photo is an example of Bonaventure Island in Quebec, which hosts a large breeding colony of northern gannets. And this is just an example of what that bird looks like on its nest. And they have these really striking light blue eyes, which will become important on the next slide. But globally, mass mortality was detected associated with highly pathogenic avian influenza at 75% of our northern gannet breeding colonies. So they breed on the east coast of Canada, but also in other locations in the North Atlantic. In Canada, as much as 40% of the breeding population was lost in the spring and summer of 2022 following the introduction of these viruses. So this is a population where it's been really important for us to continue monitoring over time to understand whether the population is recovering, what their breeding success is, whether what percentage of the population has antibodies and is likely to be protected if the virus is introduced again. And one of the interesting things that we've noted through this work is an observation that's emerged. So as I said, northern gannets have this sort of characteristic light iris. And following the introduction of these viruses, what we actually see in some of these birds is that their irises get these dark, either dark patches or completely turn black. And so it's this really striking visual feature. And so there's a couple of different things that this brings up. The first is, of course, understanding whether there's any impacts for the birds associated with these changes. So are these birds still able to reproduce? Are they still able to forage and find food? And so that work is ongoing with partners in Canada, but also across colonies in the North Atlantic. But the other thing it brings up is that could this be a way that we could monitor these populations from a distance? Because of course collecting a blood sample means we have to catch the bird, we have to take a blood sample, and for species that have been heavily impacted, that's a disturbance to them during sort of a critical time of year when they're breeding. And so we're also looking at whether this can be a way for us to monitor these species from a distance to understand what percentage of them have been exposed and survived the virus. And so this work is still underway to understand this. And now we're going to come a little bit closer to home. So, and this is a more recent example as well. So in spring 2025, we had a pretty extensive mortality event in the Great Lakes and Caspian tern. So again, another colonial breeding seabird. Mortality events were detected in multiple colonies, and these are shown on the map and in the list here, between sort of late May and sort of mid-June, so in pretty close succession. And we're still working up the exact numbers from this, But reported colony losses right now range from between 20 and 90%. So the colonies that are in Hamilton Harbor are actually the ones that experienced, to our best estimate at this time, 90% losses last spring. So very concerning. And this is another population where it's really important that we're going to continue to monitor. So how many birds are going to return to these colonies, and what do the other colonies look like in this spring and summer? So we do have planning underway to figure out the best path to monitor these species and understand the impacts the following breeding season. But another one of the activities that we do, and I said I would mention some of the other activities, is actually respond to some of these large-scale outbreaks. So this is an example of me and a colleague. We work out of CCIW, and we had reports of more sick and dead Caspian terns. And so we put on our PPE to protect ourselves, because of course it's possible that we could be exposed to these viruses. So we want to make sure we're protecting ourselves. But we went out to these colonies and we collected dead carcasses, both to submit them for testing, but also to remove them from the colony to try and prevent the virus from persisting in the environment, and trying to decrease the risk of remaining Caspian terns from getting exposed, but also other species that were nesting in proximity from getting exposed. So interestingly, during this mortality event, if you're not familiar, the Caspian terns nest in quite close proximity to a number of other species, but in particular herring gull. And we only noticed, to your point, 1 or 2 dead herring gulls on the colony and 90% Caspian tern mortality. So there is a clear difference in susceptibility between species, and that's some of the work that we're still undertaking to better understand that. I've spent a lot of time talking about birds because that's what I study, but mammals are also susceptible to these viruses, and we do see patterns in what mammals are the most commonly detected. So again, those scavengers, those predators, and largely these detections are spillover events from wild birds that have been infected and then get predated or scavenged by mammals. So this is just a list of the mammals that we've detected in Ontario. So wild mammals in the top, but I did also want to highlight some domestic mammals as well. So actually in Ontario, in Oshawa in 2023, we did unfortunately have a detection in a domestic dog that had gotten into a goose carcass that had died from the virus, and unfortunately that dog was euthanized. It did not make it. And subsequently, we've had another case in a domestic dog in Alberta, and we've also had domestic cats susceptible as well. So these cases, fortunately at this time, are still pretty rare, but it is just a reminder for all of us in the room that our pets are susceptible to this virus, and to try as much as possible to avoid direct contact with ourselves and our pets from any sick and dead wildlife that might be in the environment. And this is just an example of a multi-species interface. So we have a seal carcass here, we have some gulls that are scavenging, and then off in the distance we have a coyote waiting to come in and have a meal. And so these shared food sources and scavenging events bring multiple species into contact, creating opportunities for the virus to really jump back and forth between species. So just an example to really sort of bring that message home. Okay, so we've talked about wildlife quite a bit, but I would be remiss not to mention the impacts that are happening also in other sectors. So I'm not going to go into the details here. I do have some links later in the deck if you want more information, and I'm happy to answer questions. But we have had extensive impact in our agriculture industry, in particular our poultry industry in Canada. So millions of domestic birds have been impacted by this virus since it was introduced in 2021. And luckily rare, but we have had human cases a human case in Canada associated with this virus. Yes, I saw a hand go up.
Yeah, sorry to jump in. I just wanted to ask, other birders might have experienced this, but like in Southern Ontario, there's there's several areas where there's sort of well-known poultry production.
Mm-hmm.
Those areas are often frequented by birds, like ravens and eagles and coyotes, and it seems like some of them are still somewhat irresponsible with their waste. Like, can you comment on this? Like, I would think that it would be very strict, like, handling of poultry waste at this time. But it seems like anytime I've sort of bird at those regions, they're just surrounded by bald eagles and ravens and coyotes.
Yeah.
Like, it's sort of frequent,
Yeah.
irresponsible.
Yeah, and so that is a concern, and I couldn't have asked for a better segue. So, what's really important for some of the research that we do is understanding these interfaces, right? So where virus is spilling over and back from wildlife into poultry and then back into wildlife, and also the interface with humans as well. So I have a couple of slides just talking about some of the research that's currently underway. And this first one is speaking exactly to that question. So in Ontario, we have this great One Health collaboration. So One Health means an issue that affects the health of the environment, of animals, and of people. And so avian influenza is a great example of that. And for issues like this, it's really important that we bring teams together with expertise across these sectors so that we can really address these complex issues. And so this is a great example. It's funded by OMAFA, which is part of the provincial government in Ontario, but we have a lot of different partners involved in this project. And what we're doing is we are studying exactly that. So this is just like a make-believe poultry farm, but it's to show that we're combining our surveillance data. We have cameras set up on poultry farms. We have song meters. We're doing surveys to do exactly that, characterize what species are frequenting poultry farms, what areas of the poultry farm they're frequenting, their behavior, to try and identify ways that we can mitigate that spillover and spillback risk. And so this work is still very much underway, but the goal is to support both agriculture and the conservation of wildlife. So wildlife can be at risk if there's an infection on this poultry farm and there's waste or associated things in the environment and wildlife are interacting with that can be a risk. But similarly, if there's wildlife frequenting that poultry farm that are infected, we can see spill over onto the farm as well. So the goal of this project is to better characterize this interface between wildlife and poultry farms to try to identify ways to mitigate this spillover and spillback. And then a couple of research projects that are underway to better understand the human interface. So on your left-hand side of the screen, we have a project in collaboration with the University of Guelph, or actually led by the University of Guelph, where we're trying to understand how migratory bird hunters access information about avian influenza, what information is interesting and useful for them about avian influenza, so that we can inform the work that we do and how we communicate about the work that we do. So if you're a hunter or you know someone that's a hunter, this sounds relevant and interesting for you, there is a QR code there where you can participate in this research to help us learn more. And then on the other side of the screen is a project that's in collaboration with the University Health Network in Toronto to better understand the exposure risk for people that are working either as part of their job or as a hobby with wild animals or domestic animals, to try and understand exposure risk in those human populations. So again, if either of those sound interesting or relevant, I'm happy to chat with you after, or you can access information at those links. Yes?
Do you partner with Ducks Unlimited?
Yes, we do. We do have collaborations with Ducks Unlimited and also Delta Waterfowl as well. We've done some studies where we've deployed trackers on domestic ducks and sampled them for avian influenza. So trying to better understand whether when they get infected, they're apparently healthy to us. We can't see any obvious signs of infection, but does it change their movement and their migration patterns? So yes, we do have collaborations with Ducks Unlimited and Delta Waterfowl. Yes.
So the question hopefully will be like helping you, but typically when I encounter
Yeah. Thanks for the question, and it's like I planted you folks in the audience. I swear. So I have a couple slides with some take-home points, and the first one is about exactly that. So what does a bird that is sick with avian influenza look like, and what can we do about it when we see them? So I have 2 videos that show clinical signs of a bird infected. So before I play them, I just wanted to let you know that. They're not very long, but if you're not interested in watching them, you can avert your eyes and I'll let you know when it's over. So that one doesn't look like it's gonna work. Oh, I'm not sure why that one turned black, but in any case, so what we can see is this is actually a northern gannet, and it's showing very neurological signs. So they can have tremors, they can be circling. I'll play it again for folks. Maybe this other one will play. No, it's not gonna let me do it. Okay, play it again because it was kind of fast. That one's not gonna work, sorry. But it's another example of sort of a neurological bird. And so this is quite common for birds that are infected with avian influenza. We can also just see a lack of energy or movement that would be atypical for that species at that time of year. So it's not always as dramatic as what I showed in the video. You can see swelling around the head, neck, and eyes. That's obviously harder to appreciate from a distance. But typically what we can see is actually these birds just found dead, and even wild mammals just found dead, and typically they're in good condition because what we note is that the virus typically infects them and they die pretty quickly. So it's not like they're emaciated in most cases. That's not to say a bird that's already emaciated can't also be infected with the virus, but these are just general patterns. So the recommendations is that if you encounter a sick or dead wild animal, not to touch it and to not let your pet interact with it as well. And we actually recommend that you report it to the Canadian Wildlife Health Cooperative in Guelph, and the phone number's there. There's also the QR code here, which provides that contact information as well, but also more detailed information. So when you call the Canadian Wildlife Health Cooperative, they may ask you to collect that carcass if you're comfortable, or somebody else might be sent out to collect it if you're not, and they'll outline a safe way to do that. So that information is also available at this link here. But it may be the case, remember I said we don't test every single individual that dies. If they're already aware or have confirmed the virus in that area in wildlife, they may ask you to dispose of that carcass if you're comfortable as well. And so they'll walk you through how to do that. So thanks for the question. We do ask that you report it. And actually it's really important, and we rely on folks like you to tell us about these signals that are out in the landscape in our wildlife, because we don't have people on the ground everywhere. So it's really important for us to get that information. The other take home, and I was going to wait a second to see if somebody else would ask me, which would lead well into this slide, but I'll just jump into it in the interest of time. We get questions about, are there risks associated with feeding birds in your backyard? And as we talked about earlier, songbirds are typically considered lower risk. And so the use of feeders is unlikely to spread highly pathogenic avian influenza virus, and the risk of an outbreak in wild bird species that frequent feeders is considered low. However, the risk isn't zero, right? And so there's just a couple of things to keep in mind. So if you're in a location where waterfowl or gulls or other species are visiting your feeder, that does increase the risk. And if you have poultry beside you, or you raise poultry, it is recommended to not have bird feeders or other wildlife attractants that would bring wildlife and those poultry into proximity, because that can increase risk as well. And then just general guidance is to make sure you're cleaning your bird feeder regularly, but that's not just for avian influenza, that's for multiple other pathogens as well. And again, there is more information at these QR codes as well if you're interested, and I haven't covered something there. Okay, and so just leaving you off with your important role, and I've talked about this a few times, but reporting sick or dead wildlife, we really rely on folks like you. We really use that to know where to look and to get these samples that tell us about what the virus is doing on the landscape and how it's impacting our wildlife. Also participating in citizen science and these research projects that I mentioned. It's really important that we get your perspectives, and so we know how to communicate and the information you folks are interested in learning about the virus and the work that we do. But then also just more broadly, this first point, and I think something maybe we don't talk about enough, is supporting positive and balanced narratives about wildlife. So for the past few years, there's been a lot of talk about the risks associated with wild birds and avian influenza viruses, and all of that is really important to know and to be aware of, and we've talked about some of that today. But sometimes the message can be too biased in that direction, and it makes people worried about the presence of wild birds on their properties or in the parks that they frequent. And so it's also really important that we continue to talk about, and you folks are really good at this, the important role that these species play towards healthy ecosystems and the reasons it's important to have these species continue to be present and to conserve these species. So I think that that's an important role that we can all play as we continue to deal with the impacts of this virus. And so with that, I'm going to wrap up. I promised I would bring these resources back, so if you didn't get a chance to access them before, please do go ahead. And this video is us releasing a bird at Hawkcliffe after it was banded and sampled for avian influenza. So happy to take questions with any time that we might have left. Yes.
It's not a question, that's a compliment.
Okay.
I'm very impressed with your mastery of your subject and how well you transmitted it to us.
I appreciate that. Thank you. Oh, I'll let you direct the questions.
Thank you very much for that very interesting and informative talk. I just wanted your brief opinion on that very famous yet tragic incident that occurred on the West Coast last fall with the ostrich farm. Two very opposing viewpoints. One, I guess we had to call them all. The other, well, it's been so long since the others died, so maybe they have developed immunity.
Yeah. Yeah, so that was a really complex issue, and I want to be really transparent that that's outside of my day-to-day role. So that's colleagues at the Canadian Food Inspection Agency that are responsible for managing virus on premises like that. But it was really, really tricky, and I work in close collaboration with a lot of those colleagues, and so there was a lot of considerations that went into that, both on the side of sort of upholding the rules, and this is how we get rid of the virus from the landscape, which is what we're trying to do, right? We're trying to mitigate and eliminate it, and these rules are in place in order to help us and facilitate us doing that. And so I do just want to acknowledge the hard work of the colleagues that do that work. It's a hard job. And I do know there were strong views on the other side as well, and I won't really comment further on that. It was a really challenging a topic, for sure. Yeah, and it did get a lot of media attention.
Yes, you sort of touched on this topic when you mentioned about bird feeders.
Mm-hmm.
Like it not being like a significant source of transmission, and to like clean them regularly. But what about like this being like an urban like area? What about like water that you put out for animals, right? So like I leave water out because there's no like wetlands and that, right? So if you leave like when I leave water out, it's like a big bowl of water. There'll be like birds in the summer, like regularly like bathing in it. Then squirrels will drink from it. Then possums. Then raccoons. Then stray cats, right?
Yeah.
Like overlap. So I mean, it's just in like one day that would happen, right? So it's like, would I be like cleaning this like bird bath / water source like every day, or like what would be an appropriate response?
Yeah, that's a great question. And if we're talking specifically about avian influenza, Avian influenza can survive in the environment under certain conditions. So it doesn't really like really hot conditions, which is why typically we don't see a lot of virus in the summertime. It doesn't like really salty conditions. There are sort of, there's more nuance to it, but in general, avian influenzas can survive in the environment. So in the instance that there was an individual that was infected with the virus that visited a water source like that, it is possible that that could serve as a way for the virus to move between species that may not otherwise come into contact with each other in that way, right? So as with bird feeders, the recommendation would just be to really try and renew that water source as often as you can. And if it is a moving water source, that can decrease the risk as well. But the risk is never going to be zero associated with that for avian influenza and also for other pathogens as well. But you do also have to balance that with the benefits of providing that type of resource for our wildlife.
Yeah, so it's not like disinfecting, it's just dumping it in the water.
Yeah, yep. And also, it can help to stay aware of what's going on in your area. So there is a public facing dashboard, which is the second QR code here, which actually tells you about where we've detected virus, both in wildlife and in poultry. And so you can keep an eye on that to see what the activity is like in your area. If there's a lot of virus that you're seeing a lot of detections in your area, and you can zoom in on the map, not super close, but you'll get an idea of generally your general area. If there does seem to be a lot of activity at that time of year, that could be an opportunity to maybe take that source away until things start to peter off again. Yes.
Sorry, this question.
Oh, sorry. I'll let you.
Yeah. In cases of what are often called crowd diseases, over time, the mortality rates that are initially really high go down.
Yep.
As well as the extents, the actual impact of the virus on the population. Is this the case with the populations you're talking about? So the gannets, the initial experience was really severe, but in follow-ups, has there been similar high mortality rates like this, or with Caspian terns, or is this a situation where one can expect over a period of time that birds that are encountering this virus for the first time in large populations may suffer greatly,
Yeah.
but that over time the antibody rates go up and they learn to live with the virus in some way, and the virus learns not to kill its food source.
Yeah, great question. And that's really what we're hoping for. And in the specific gannet situation, we haven't seen extensive mortality events in follow-up years, which is really great news. But what we're trying to clarify is whether that's because the virus hasn't re-entered that population, or whether it's because those individuals have immunity against the virus. In the Caspian terns, we don't know yet because this was just last season, and so we're going to wait to see what happens this season and monitor them closely. But what we're hopeful for is that the birds are developing these antibodies and it will protect them against reinfection, but we're still learning about how long these antibodies persist in these species. And the other thing I will say, I didn't cover this slide because I didn't have time, but viruses are in the business of changing, as we learned with SARS-CoV-2, right? And so what this is showing is the number of different forms of the highly pathogenic avian influenza virus that we've been talking about over time. And so we can see that it's not just one virus, and it's quickly changing. So there's a lot of different viruses, and it tends to increase pretty dramatically over periods of time when birds are at increased density. So the virus is moving around and transmitting more, increasing opportunities for that virus to be able to mutate and change. And so the other thing we're still learning about is whether as the virus changes, the antibodies that they generated against old versions of the virus will still protect them. And so there's a lot of unknowns there, but what we're really hopeful for is that, exactly as you say, as these populations get exposed, they do develop some degree of immunity that will protect them moving forward. And that remains to be determined, and it's why it's so important that we continue to monitor these populations over time. Yes.
Can you tell us anything about the snowy owls that have been found out on LSD streets this winter?
I can't tell you about those specifically, but snowy owls have been heavily impacted by this virus. We have had a lot of detections in snowy owls across the country, and it is something we're looking to do moving forward, is to find opportunities to screen this species for antibodies as well. We haven't done any of that yet, but it's really important that we do understand what proportion of those individuals are getting exposed and surviving. But snowy owls are definitely susceptible to the virus. Yeah.
Was a rise in bird mortality related to our COVID? During those years, did they have a rise in bird mortality?
As a result of COVID, do you mean?
Not as a result.
Okay.
We had COVID, but I wondered at the same time, was there a rise in bird mortality from their avian influenza?
Okay, good question. Yeah, because I was going to clarify that as far as we know, birds weren't susceptible or impacted by COVID. But the other thing that COVID made challenging, obviously there's a number of things that it made challenging. But for those conducting field work, they often weren't able to do that field work during those years, and so some of the monitoring data is not available from those years. But to answer your question specifically, largely during the peak of COVID, highly pathogenic avian influenza wasn't here yet. So it wasn't here until the end of 2021, and that was just on the East Coast. So it wasn't in other locations until mid-2022, and depending on where you were in the country, sometimes later than that. So not as a result of avian influenza, but I can't speak more broadly to whether there was other causes of mortality increase during that time. And there's probably some issues related to data that we weren't able to collect over those initial years as well.
We're running low on time. I'll get one more there. I was going to add a question.
Yeah.
Because I saw the northern gannet thing in the eyes. Has there been any monitoring of just photographing existing gannets to see if you can detect the virus that way?
Yeah, so we can't detect the virus through photographs, but we can detect these iris color changes. And so we actually have a project right now where we have a student developing an approach from a photograph to actually quantify how much of a change in iris coloration there is, and then we're pairing that with our antibody data to see what association there is between the extent of iris color change and antibodies, to better understand whether we can use that as a way to screen these populations for exposure. So yeah, it's a really interesting finding that came about, and there are a number of research projects underway related to better understanding that.
You just briefly mentioned this previous answer, but earlier in yours you had a slide, lots of bar graphs, pink and orange bar graphs, and over about 4 years showing the prevalence of I think it was the highly pathogenic. But I'm just curious, that's the one. I'm curious, is that percentages on the left, or because I'm wondering whether sampling effort has been uniform over that whole time frame, or I assume sampling effort hopefully has been ramping up as it's become more prevalent.
Yeah. Thanks for the question. It's a great point to clarify. This is not percentage. This is not prevalence. This is just the number of detections. So both of these represent, it gets into a little bit of nuance, but the red are confirmed highly pathogenic avian influenza, and the orange are suspect. And that gets into the nuance of some of the diagnostics, but no, this doesn't show sampling effort. And the reason I didn't put that on this slide is because the sampling effort dwarfs the number of detections of positives. So we have been doing an exceptional amount of sampling since fall 2021. And over the years, it's gotten more targeted as we've learned more about what species the virus is most likely to be detected in. So yeah, that's a good point. This does not represent prevalence.
We'll take a couple more questions and wrap up.
Does the Irish discoloration only occur in gannets?
Something else we're also trying to understand. So in species that would normally have a dark coloration in the eyes, it's unlikely we would detect a similar change like that. But we do have a study in gulls as well, and there's been some reports of iris color changes in gulls. And so we have a project underway as well to do a similar study, as I mentioned, for the northern gannets, to see if there's an association there with exposure to the virus.
It seems like this avian influenza seems more prevalent in the last number of years. What's the reason for that?
Yeah, so great question. Do you mean in Canada or globally? Or both? Okay, so in Canada, it's definitely been more present over the last few years because we've never had it before, at least this version of the virus. So this highly pathogenic virus was newly introduced to Canada. Our birds and mammals were naive to this virus. So certainly the activity of this virus has dramatically increased since then because detections before that time were zero. But globally, even in areas where the virus was present prior to when we had it here, the activity of the virus has been increasing as well. And we're still working to understand why that is. But as I said, viruses are in the business of changing over time. And so likely it's related to the virus's continued ability to adapt to different species and to wild birds. Yeah.
On behalf of the Nationals Club, thank you for informing us about this really important subject. If anybody has any further questions, if you've got a few minutes to hang around after.
Yes, I do. Thanks for having me. It was great to be here.